Hello everyone and welcome to my poster. My name is Marius Mihasan. I'm from Alexandrian Cuza University of Iași, Romania. It is my pleasure to present to you some details about why and how we print physical macromolecular models in my lab. In the general chemistry and biochemistry lectures that I teach, my main focus is to make students understand the structure to function relationship that governs biology. I have noticed more and more that biology students, albeit able to recognize chemical formulas, not fully understand their significance. Important aspects, such as atom size and shape, bond length and symmetry, and generally a sense of spatiality and dimensionality, are lost. Physical models of molecules and macromolecules can improve that and offer a far more complete learning experience. That's why we have started using such models in our teaching. The models that we are using for teaching and demonstration need to be based on real scientific data, and we are happy to have PDB here, which offers such a wide variety of structural data on so many macromolecules. These models need to be depicted using standardized representations, and nowadays we have really good free software which achieves that in a very intuitive way. Also, these models need to be easy to edit and adapt to the specific outcomes of a lesson, need to be cheap to fabricate and reproduce, and easy to distribute. And luckily, we have 3D printing, which fulfills all these three targets. 3D printing technology allows for the fabrication of a physical object starting from a computer-generated model. 3D printing technology has reached a level of maturity that allows for a rather reliable printing experience and also rather low costs. Since the start of the pandemic, I have worked on putting together a method that would allow the fabrication of macromolecular models as cheap as possible using 3D printing. The protocol depicted in the second part of my poster here relies on a under $500 printer and uses the cheapest PLA that I could find. Basically, the protocol involves three main steps: generation of the computer model, printing of the model, and then cleanup of the model and sanding and painting if required. The first step starts by downloading the atomic coordinates of the molecules of interest from PDB and choosing a visualization style based on my needs. I then edit the rendering parameters of the model to improve the printability and save the file as an STL file. Next, this STL file that contains the computer model is it's opened in a slicing software where the printed scale is set up, as well as some other printing parameters, including the generation of the support material. Thus, this support material is some extra plastic which is added to the model in order to allow the printing of some pieces of the model that would not print otherwise. The slice model is then converted in a G-code file which contains instructions on how the printer should actually create the physical model. After printing, the model looks somehow like this. This is a freshly printed model of insulin, and this part white here, this part white here, and here, it's actually the support material which needs to be removed in the final step of the preparation of the physical model of a macromolecule. The last picture here is the final model of insulin where the support was successfully removed. Following these steps, I was able to print more than 40 proteins in DNA models that I currently use for teaching. All these models are available for download and for printing if you follow the link which is available in the abstract file associated with this presentation. Some representative models are presented here. In figure 1, for example, it's the physical model of an ATP synthase in which every component of the ATP synthase can be removed and manually placed back in the complex. The whole complex can be assembled and disassembled in front of the students. In figure 2 are the physical models of two antibodies which are interacting with the same antigen, which is lysozyme depicted in white here. The light and heavy chains of the antibodies were printed separately in different colored PLA and then glued back together. One of the antibodies also has the sugars, which were also printed separately in gray PLA and then glued inside the heavy chain. By far the most complex printing project that I have done is the complete replication fork, with all DNA, RNAs, nucleotides, and all proteins involved in the process. Each component, each color that you see in figure 3 was printed separately, and then the components were glued back together. In order to achieve this, the DNA and RNA was printed in flexible TPU. More technical details on how to render molecular models in order to have successful prints, including a set of printing settings that work, which focus on low cost, can be found in the paper in this link here at the end of my poster. The paper was published in Biochemistry and Molecular Biology Education, and I'm really happy about this. Thank you for your attention. I will be more than happy to answer to any of the questions.